Title of today's presentation is Alternative Splicing, or AS Theory of Aging and its relationship with COVID-19. So the background is this: Alternative Splicing, Theory of Aging. The figure is borrowed from this paper over here, Systematic Transcriptome Analysis Associated with Physiological and Chronological Aging in C. elegans, published this year. The physiological aging referred here is actually meaning the biological aging, not the chronological aging. And what they found was that there is an increased usage of distal 3' splice sites as the biological age increases in C. elegans. And as you can see over here in the cartoon figure, the proximal site is being less used, and the distal sites are being more used as you age biologically, and the resultant gene size gets smaller. So that's one finding about alternative splicing in aging. There is abnormal alternative splicing in COVID-19. As you can see over here in the enrichment plot, the top enrichment was found to be associated with RNA splicing genes. And over here, as you can see, this is inferred cell types. This is not the single cell analysis, but bulk analysis and deconvolution, giving you the different cell types. And you can see there's different splicing activities going on in different cells. And it seems that they are aberrantly regulated when the COVID-19 comes. So in this paper, they found that bulk RNA sequencing identified 1,383 genes, and these have the differential transcript usage DTU in COVID-19 patients, and these were found to be associated with coagulation. And for other paper, which is not included here, they found that in lungs, not blood, they found that the alternative splicing, rather than the total RNA expression of these genes, were actually associated with COVID-19 severity. So saying this, what we want to argue is that first is that there is alternative splicing being changed when aging, as well as COVID-19, and we want to actually see if there's any interrelationship between them. Methods, we're thinking of using the lift cutter, Fraser, and die-cast. So lift cutter is published, it's been out there for a while, but it's actually been used by the GTEx consortium, which gathers all the expression public data in American ancestries, and they've used this, so the tool is credible. And more recent tool is called Fraser, and this is actually updated by the DROP pipeline, the detection of RNA outlier pipeline. So this would be useful as well. And last of all, the latest one is die-cast, which ASTA is working right now. So we want to first of all look at the leaf cutter and go down if we can get the common signatures from all these different tools. So these are the possible trinity of alternative splicing, COVID-19 aging is going to be investigated, and we want to actually construct DB, database of age-associated AS transcripts, called HASDB, and we also want to actually make a pipeline out of it called biopipe transcriptome. So we want to integrate this into our existing pipeline, but the problem is that there are a few limitations, including that we only have short read RNA sequencing samples available, and normally alternative splicing requires long reads, and we only have whole blood and PBMC, there's typo PBMC samples, and AS, or the alternative splicing, is known to be highly cell type specific. And one of the major problem that ASTA has encountered before was that all of the tools support only HG19 or GRCH37, so this is problematic. One possible solution is to actually lift over from the 38 to 19, but this is not good. We can have duplicate reads, errors, wrong annotation according to ASTA, And it's better off that we start off from the scratch using HG19 as the reference to map our RNAs. So these are the designs. We are first going to align the WGP RNAs to the HG19. We're going to stratify the samples by age, sex, COVID-19 severity. We're going to do the correlation analysis, but we haven't decided which variables we're going to use. We're going to define what physiological aging is. Actually, we have to find out what physiological aging, the definition of it in the first paper. And our hypothesis is that physiological aging is associated with increased usage of distal 3' splice sites, obviously, particularly in known age-related genes possibly, and we could also suggest new age-related genes. C. elegans and human compared to biology could also be overlooked by aging. And we could also possibly use GEO, or the public database, which collects all the RNA experiments, to compare differences in splicing by ancestry. This is a good suggestion by Esther. The next meeting is going to be held next week, on 22nd of December. Thank you.